students. So I am here to test your knowledge this time. So I hope you all have revised the biomolecules, and today we are going to discuss a lot of questions. That means what we have prepared on the basis of NEET pattern. So it would be pretty simple as compared to what we have in IIT JEE and mains. So let us just have the exercise for that. Now the first question says, which of the following statements is not correct? So I need to find out, I need to analyze each and every statement for that. So let us just have the first statement. Insulin maintains sugar level in the blood of human body. So we all know that insulin actually maintains the blood sugar level, that is a hormone. So we all know that. I need to find out what is not correct. So just to read the question carefully every time. Ovalbumin is a simple food reserve in egg white. So if you do not know what ovalbumin is, you know what albumin is. Albumin is what? The white portion of the egg. So that is what the statement is saying. That means it must be correct. Let us analyze the another one. Blood proteins thrombin and fibrinogen are involved in the blood clotting. Yes, we have done this very kind of a concept in the importance of proteins. So that means this very thing is also correct. Now let's move on to here. Denaturation makes the proteins more active. Is it so? So just try to recall the concept. Denaturation is actually entangling of the protein. That means here what we have got? Here is a protein. That means so many kinds of structures, primary, secondary and tertiary, which are actually entangled into each other. That is what a protein, a proper protein is. And denaturation means when this very thing, due to temperature or pH changes, it actually entangles, it actually opens up. So that means we have got something like this. This is something, a denatured protein. So if you take an example of whenever you take up milk and you add some lemon juice into it, what is happening? The protein that is present in the milk actually has got this kind of a structure, and whenever we put the lemon juice, it loses its activity, it loses its biological properties and getting denatured. That means it then coagulates. It forms a structure like this. That means it gets entangled. So as per the statement over here, denaturation makes the proteins more active. It is not so. It is actually making the proteins more inactive, making them lose their biological activity. So they actually lose biological activity. So I hope you all have got it. Let me just mark the answer right. So that is the answer over here. This very statement is not correct, otherwise all the three are correct. So that is what we need to find out. So let's move on to the next very question. What does it say? So the next question here is, in a protein molecule, various amino acids are linked with. So it's a pretty simple question. We just want to know the linkage. Protein molecule has got so many amino acids and how they are attached. So we have done this very procedure. So they are attached with beta glycosidic bond? No. Peptide? Yes. It's not a dative bond. It is not glycosidic anyways. It is always a peptide bond. Why this is so? So let us just try to have a very simple amino acid. So how actually the two gets connected? So I need to form gly-ala. That means glycine separated by alanine. So I've got glycine, that will be, let me just make this again, CH . So that was the part for the glycine, and this is the remaining part is for the alanine. So we have formed the gly-ala. So that is alanine, ala and gly. Where is the peptide bond present? Here is the peptide bond present. So that is the peptide bond. That is how the different kind of amino acids link onto each other to form a bigger molecule, that is a protein. or we can say polypeptides. So here it is the answer. So next one says what? So let me just rub this off first. Now it is asking a very simple question. This is the correct statement regarding RNA and DNA. So we need to have a proper knowledge of what RNA and DNA is. The sugar component in RNA is a ribose and the sugar component in DNA is 2-deoxyribose. Is that so? I need to find out the correct statement. Let us just analyze the rest of the three. The sugar component in RNA is arabinose and the sugar component here in DNA is ribose. So they are actually asking about the components in RNA and DNA. So let us just have the full form of both of them first and it will give our answer. What is RNA? Ribose nucleic acid. And what is DNA? It is 2-deoxyribose. So that is, I can say deoxyribose followed by nucleic acid. So that is the full form and it gives our answer. So basically what we have got is pentose sugars involved in both of the structures, and just only the full forms have cleared our doubts, have given up the answer, that is RNA is actually ribose and DNA is 2-deoxyribose, and none of the options containing, because every other option is actually containing arabinose, which cannot be the sugar involved over here. So I hope you all have got the right answer here, that is A. So let's just move on to the very next question. What do we have next? So the next one says, which one given below is non-reducing sugar? So how do I find out a non-reducing sugar? So basically a non-reducing sugar is the one which is having either the aldehyde group or the ketonic group present. So what is a non-reducing sugar? It is having aldehydic as well as a ketonic group. So either of the two groups available for the reaction. So it must be having CHO or a carbonyl group available for reaction. That is what is known as, the reducing sugar. That means it is giving up the reaction. So that is a reducing sugar. And I need to have a proper idea about the structures of all the four given in order to find the non-reducing one having no such kind of aldehydic or ketonic group available for the reaction. So one is lactose, the other is glucose. So what is lactose first? Lactose, we all know that is beta galactose. So it's beta D galactose plus beta D glucose. So that is what lactose is. Here C1 is connected via C4. What is glucose? We all know glucose. That is what? Having CHO free, that is aldohexose. So that is aldohexoses and it has got CHO over here. Now in the sucrose what do we have? We have got glucose plus fructose. And glucose here is alpha D, and it is beta D fructose. What is maltose? Maltose is again actually having linkages like it is glucose. What is alpha glucose? And again an alpha glucose connected via C1 and C4 linkages. So if C1 linkage is connected with C4, that means where is the aldehyde group present? That is towards C1, and this very molecule will be involved onto the C4. That means C1 is still free for this alpha glucose. So here C1 is free. That means the group is free. That means this is reducing. So I hope you all are getting the point. So So you need to dig into the structures for that. Sucrose, alpha D glucose and beta D fructose. So let us just have the structure for that. I have got there will be a beta glycosidic linkage here. So that would be something like this. So it would be somewhat like this. This would be the structure. The rest of the things I am not completing because we all know what all the other groups are present over here. So that is alpha glucose and that is beta fructose. So you may be having an idea and you need to complete. I am not spoon feeding you. So this is what kind of a linkage here is. So both the groups over here are occupied. That means no reducing group present. And what do we have in this? Glucose is definitely a reducing one. And what about beta D galactose and beta D glucose? Again, C1 is linked with C4. That means here C1 is free for the reaction. That means this is also reducing. So I hope you all have got the answer by now. That is sucrose, where both the positions are occupied, so there might not be any kind of a reducing reaction over here. That means any kind of reaction, that means which is concerned with the Felling's solution or which is concerned with the Tollens reagent. So that is not there. So I hope you all have got the answer here. That is sucrose. Sucrose is the only non-reducing sugar. Now let's move on to the next. So what do I have here? Very simple question again. So it looks complicated, it looks confusing, but it is not. The correct corresponding order of the names of the 4 aldoses with the configuration given below. So they have been given the configurations. I need to have the names for each one of them. So if you remember, what are these aldoses? So these are 1, 2, 3, 4. That means aldotetroses. So what have we discussed in the case of aldotetroses? I have got erythrose and I have got threose. So what is erythrose and threose? So let us just draw the structures first. So that is erythrose when both the OH are on the similar side, CHO, since this is aldo and CH2OH, that is the remaining. In the threose, what do we have? We have got OH arranged opposite. So that is what it is. That is the structure for threose. Now I need to find out the configurations as well. That means they are asking about the configuration, that is D and L. That would be correlating with the glyceraldehyde. So if the OH, so this is what? This is erythrose and this very OH, this whole thing, OH is lying on the right hand side, that means it will be D-glyceraldehyde, so that is D. If I move on to here, what do I have? This is nothing but the threose. And this very, again the OH is lying to the right hand side, that means the D-threose. What do I have here? I have got, again this is nothing but an erythrose kind of a structure, but what do I have? I have got this as the left hand side, that means the L-erythrose, so this will be L-erythrose. And what next? I have got this is again but L, so that is L-threose. So what do we have? We have got, do we have an option here? D-erythrose, D-threose, D-erythrose, D-threose and L-erythrose, L-threose. So that is what we want. You can see over here we have written. So D-erythrose, D-threose, L-erythrose, L-threose. So that is our answer. You can also check the rest of the options, and we have got the answer over here, that is D. I hope everybody has got it. So now let's just move on to the next very question. What do we have? So let me just rub this off first. Now it's again a very good question. Also it is very important because here they are asking about the formation of oxime. So what does it say? D plus glucose react with hydroxylamine, okay, and yields an oxime, the structure of the oxime. So let us just first have the reaction. So they are actually wanting the structure. So this is the D plus glucose. Let us just make it out. So glucose is aldo hexosis. That means 6 carbon atoms. We have got 6. OH, OH. So what is that is the structure of D plus glucose. D for this OH is lying on the right hand side. So that is what we make. And I need to react it with hydroxylamine, that is NH2OH. What do I get? I get a simple reaction formation of oxime. So how does it actually take place? How does the reaction take place is, and this is NH2OH negative. So it would be something like this. All the other groups will remain as it is. The changes would be only in the aldehydic positions. So aldehydic groups will be changing. That is, it will be. So that is what I need. This is nothing but oxime. So that is nothing but oxime. So let us just correlate it. So I hope you all have written this down. So I am rubbing this off because now here comes up the options. So let us just have the options here. What do I have? In the first very structure, so just try to compare it with what we have made here. This very thing over here in each of the options they have given. Now let us just compare the rest of the structure. So the rest of the structure will be something like what we have made, that is an actual structure of glucose, OH to the right, then left. No, this is not done over here, that is galactose over here. Also this very structure is not our concern. If you see over here, yes, definitely that is what we have made in the case of glucose. So just be very careful how the configurations of OH are arranged. So that is our answer. D is the answer over here. You can have a check over here. This is not our option. So I hope everybody is clear with the concept. It's super easy question. It needs actually reaction mechanism, the formation of oxime. You must have done this very mechanism in the case of aldehydes and ketones as well. So that is just a simple reaction what we have done. Let us move on to the number 7th. So what we have? Okay, Chargaff's rule states that in an organism. So what is the Chargaff's rule? Chargaff was a scientist, so he told us about the molar quantities of the bases. What kind of bases which are present in the DNA? A, T, G, C. So as we all know that A is linked with hydrogen bonds with T. G is linked with hydrogen bonds with C. That means what Chargaff told was the molar quantities of A is equal to T. Molar quantity of guanine is equal to cytosine. So that is what we need here in the answer as well. Amount of adenine is equal to that of cytosine? No. Amounts of all the bases are equal? Not at all. Amount of adenine is equal to that of thymine? Yes, A is correlating with the T, and the amount of guanine is equal to that of the cytosine. So that is what we need as answer. So that is the answer. Amount of adenine is equal to guanine. So this is also an incorrect option. So I hope you all have got, that was a pretty easy question. You need to have the proper knowledge of it. So now let's move on to the next question. What we have in the next question? Which of the following is correct statement? So starch is a polymer of alpha glucose, that is the first statement. I need to have the correct. That means the 3 of them will be incorrect. So let us just analyze. Starch is a polymer of alpha glucose, is it? Here is starch, and actually it is a part of amylose as well as amylopectin. And we all know in case of amylose, what do we have? We have got alpha D glucose. connected via C1 and C4 linkages, and in amylopectin I have got connections of C1 and C6 beta linkages as well. That means forming a branch chain. This is just a linear chain. So I hope everybody has recalled the concept over here, recalled the logic behind starch, that is amylose and amylopectin. That is actually a group. Amylose here would be 15 to 20% and amylopectin is around 80% in the starch. So that is the composition of starch. I think I've got my answer in the very first statement only. That is, this statement is very true. But let us just have the other ones. Amylose is a component of cellulose. No, it is not a component of cellulose but starch. Proteins are composed of only one type of amino acid. Is it so? No, there are so many kinds of amino acids. Even our DNA contains approximately 400 kinds of amino acid present. So basically this option is also incorrect. In cyclic structure of fructose, there are 4 carbons and 1 oxygen. Is it so? Not at all. No 4 carbons but 5 carbons. So they are 5 carbons present, not 4. So I hope everybody has got the answer here, which is number 1. So let's just move on to the second last question here. So it's a very interesting question and I hope everybody is enjoying it. Which one of the following gives positive Fehling solution test? So we have already done this. I just told you that the reducing sugars, what we have, reducing sugars always give positive Fehling solution test. What is a Fehling solution test? So this is the Fehling solution that we are going to take and we get Cu2 plus as the red precipitate. So I need to have a proper reactant for that. So is it sucrose? No, because it is a non-reducing sugar. Is it glucose? Yes, definitely, it is the glucose. So let us just have the structure for glucose. CHO. It is CHOH. 4 times I am writing and CH2OH. So that is what it is. And what do we have? What will be the reaction and what into what the glucose gets converted? It is this COO negative and the rest of the molecule remains the same. So that is actually the Fehling solution test. Fehling's test, which is positive, which has come out to be positive in the case of glucose because this is the reducing sugar. What about the fats? They do not contain any kind of aldehydes or ketonic groups. So we are not concerned about the fats as well as the proteins. Again, there is no concern of Fehling solution test with the proteins. So I hope everybody has got the answer, that is glucose. So it is a matter of reducing or a non-reducing sugar and you will be getting the answer right. So let's move on to the very last question of this questionnaire round. What do we have next? The alpha D glucose and the beta D glucose differ from each other due to the difference in the carbon atom with respect to its. So that is the question. I have got alpha D glucose. What is alpha D glucose? So let us just make that. And I have got what? I have got beta D glucose as well. So basically the difference is at the anomeric carbons. OH may be on the right and OH may be on the left. So that is the difference. That is the basic difference. So here, let me just first have the loss of the water molecule. That is how we are going to check. OH, H, OH, H. And how do I need to join it? I need to join it via this. What do I obtain over here? I obtain a structure like this. So that is what we have in the case of alpha D glucose. And what about this? Again we get a similar kind of a loss of water molecule. This is what? This is a glycosidic linkage. I hope you all have recalled the concept here. And what do I need? I need again the carbon atoms, OH, H. So that is what we have. Here if you can see, this is beta, that means lying to the left. So this is lying to the left, this is lying to the right. That is why we call it as alpha or beta. These are nothing but the carbons over here. We call them as anomeric carbons. So we have got the answer here. Let us just check. The difference in the carbon atom with respect to its what? Conformation, configuration, number of OH groups. Number of OH groups are always same. So this cannot be the option. Size of the hemiacetal ring. We are not concerned about the size. It's approximately the same. Conformation or configuration. So conformations is basically whenever I'm trying to rotate and have different kinds of conformations. But over here, different kinds of conformations for glucose is not possible. I hope you all know what conformations is. But if you see in the configuration, what is configuration? That is concerned about the arrangement of one particular group, whether it will be right or left, whether it will be cis and trans. So configuration we have done in the case of geometrical isomerism. I hope you all have recalled the concept. So on the basis of that, I have got the configuration as my answer, but not conformation. So do not get confused between the two terms. You must be having clarity about the concept. So B is the answer over here. So I hope everybody has followed me. You need to just practice and revise all of these questions, because I have picked these questions just in order to make you revise almost all the concepts, almost all the important concepts within the chapter. So just have a look at them, go and just try to solve them on your own. So I'll be back with the next question round in the next session. Till then, you just need to revise and practice a lot. Have a good day.